Before we go on, just a reminder, there are 6 proteins and enzymes that we talked about in DNA replication, and 7 if you include the telomerase. So make sure you know those ones really well for the final exam. All right, let us go back to our PowerPoint, and today we're going to talk about genomes, genes, the genetic code. So this is a bit of a hodgepodge kind of topic. We're going to talk about a whole bunch of things that are going to be important for the next couple of units. So genes and genetic code and how to use that. And I have one problem at the very end to show us how to, something that you're going to see on the final exam. Okay, so first let's talk about what is a genome. And I think we've thrown that word around a few times, chromosome, genome, things like that. So genome is really all the DNA in something. We could be talking about all the DNA in a cell, or we could be talking about the DNA in an organelle, such as a mitochondria. And the genome, the DNA, contains a whole bunch of things. Obviously, it contains genes, but we did talk about quite a number of structural components already: the origin and replication, the telomeres, the centromeres. These are structural components. They're regulatory components we're going to talk about in a later unit. And there are other features. And there's a bunch of DNA, actually, we don't know what it does either. And so these things are very interesting. So prokaryotes, we of course have, they have a chromosome. It's usually circular and relatively small, speaking compared to a eukaryote. E. coli, you can see, for example, has about maybe 4,400 genes, somewhere around 4 to 5 million base pairs. Usually a single copy, and it's folded. Prokaryotes may also have plasmids. They don't all have plasmids, some of them do, and that's kind of like a mini chromosome, and it will have a few genes. So usually typical is like 3 to 15 genes, and it'll have multiple copies, also circular and often supercoiled, as we talked about in the lab. So eukaryotic chromosomes are usually linear, and you can have one, but not us. Only simple organisms have one. Most organisms have multiple chromosomes. So humans have 23 pairs, so 46 chromosomes. You can see right there we're looking at karyotype of Drosophila, so that's the fruit fly, and it has 4 pairs of chromosomes, 8 chromosomes. You can see 2 of them are really tiny. I don't know what those ones are all about, why they're so tiny. There's little dot ones here, like there. And they have about 12,000 genes, so not quite as many genes as humans. Maybe they're simpler organisms, but that's not always the case. Sometimes flowers and plants can have massive genomes, and they tend to be a lot less complex than animals. So there's the human genome. We have 23 pairs, so we often talk about them as autosomal pairs or sex pairs, chromosomes. So the sex chromosomes mean that's what's determining an individual's sex usually. So the X and the Y down there, you can see this person, it looks like is a male. And we're not exactly sure how many genes, somewhere around 20,000, we think. You know, we're still learning things about genes and genetics nowadays, but that's kind of the current estimate looking at the human genome. So I kind of want to talk about how big this genome is. I find this really, really fascinating, how much information is built into a genome. You take a look at that, it says 3 billion base pairs in the haploid genome. So that means your full diploid genome is about 6 billion base pairs. That's a lot of information. So what is in that information? So one way to look at that is how that compares to works of literature. Okay, so 6 billion base pairs, that's 6 billion like alphabet letters. So if you're familiar with the Bible, that's about 3.5 million. So not even close. You're not familiar with the Bible, maybe you're familiar with the Harry Potter series, which is slightly bigger apparently, maybe around 5 billion letters. So that's not even close, right? Encyclopedia Britannica, which probably nobody uses anymore because everyone is using the internet, isn't even 1 billion letters. So what are we looking at for about 6 billion? We're looking at roughly the English Wikipedia. If you look at all the Wikipedia out there, it's found in multiple languages. It exceeds 6 billion by quite a bit. And Wikipedia, of course, has many more entries than a typical encyclopedia because there's entries on pop culture and Star Wars and probably what celebrities are on Botox, and you can imagine all the sort of useless information that is in Wikipedia. So it's a massive, massive amount of information. Another way to look at this is what is in that information. And this is the part that I find super fascinating. So here's my little chromosome, kind of made a little pie chart. So how much of that actually codes for genes? About 1.5%. Right? This is the part I find mind-blowing. So that means that 98.5% of our genome does not contain genes. So what is going on in there? Well, a lot of it we don't know, but these are estimates. About 25, 26% codes for introns, which are kind of part of the genes. And we'll talk about introns and what those are. When are we going to talk about them? Next lecture. We'll talk about what introns are. A big percentage seems to be regulatory, so switches that say time to turn this gene on or time to turn it off, those kind of things. And we'll talk a little bit about regulation and how that works. I think that's topic 18. A big percentage is kind of in that structural, we don't know what it does yet. Scientists used to call it junk DNA, meaning they didn't think it had any function, but kind of we're getting away from that because we're realizing that as we uncover information about our genome, there's a lot of things that we're learning what it does. So some of it's structural, so like I said, centromeres, telomeres, origins of replication. You can imagine all sorts of different structural components that are found in our DNA and unknown. And here's the part that I find super fascinating is that about 8% of our genome are actually viruses. That's right, you have virus sequences in your genome, and that accounts for about 8%. Super, super fascinating stuff. Something I wish I knew a little bit more about, but really cool stuff. Okay, so what else can we say about the genome? So the human genome, of course, we don't just have our main genome, but we also have organelles, such as our mitochondria, that have their own genomes. Actually, in humans, that's the only other organelle that has its own genome. It's not huge. You can see it's about 17,000 base pairs, 38 genes. So this is tiny. You can see it's showing there what the genes encode for. So a bunch of transfer RNAs and a handful of enzymes involved in the citric acid cycle and involved in the electron transport chain. So probably no surprises there what those enzymes are for, because that's what the mitochondria does. But the mitochondria does have quite a number of other proteins that have to get imported, because 38 genes is not enough to run a mitochondria. So a few genes there. Chloroplasts also have their own genomes. Here's one from tobacco, and it's bigger than a mitochondrial genome. You can see it's about 113 genes, a bunch of transfer RNAs, a bunch of enzymes involved in photosynthesis. So again, no surprise, because that's of course what a chloroplast is up to. So here's kind of just a summary slide, and this is kind of one of the themes of this class, right? Understanding the differences between prokaryotes and eukaryotes. And so I made this table for you. We're much further beyond just saying one of them has a nucleus and one of them doesn't. We want to talk about other things. So you can see it says here the size of the genome. The prokaryotic genome is usually much smaller, and the eukaryotic is usually much bigger. And there are a few weird exceptions in there. Eukaryotes usually have more than one chromosome. If you're a mammal, you're usually diploid. If you're a plant or a reptile or something like that, there's a lot of other weird things that can go on. Some of them are tetraploid, some of them have 5 copies of their genome. Some of them are hexaploid. A lot of insects are haploid, and so it gets very, very complicated once you get out of mammals. But the point is multiple copies of the main chromosomes, whereas prokaryotes, you're looking at one chromosome generally. What other things can we say? So eukaryotes, because we have linear chromosomes, we have these telomeres, and we also have centromeres for our mitosis. We also have histones, although I do have this note that some archaea do have histones for packaging their DNA. So I don't know a ton about histones, other than for packaging DNA, why archaea need them, because they also have small genomes like other prokaryotes. Eukaryotes, some of our organelles have genomes, so mitochondria and chloroplasts being the most notable. What other things can we say? Well, there's a whole bunch of things we're going to talk about that are on this chart that you can come back to this later after we've covered them, and they'll probably make a little bit more sense. Like I just mentioned, these intron things. Prokaryotes don't usually have introns. I think there's a few exceptions out there. Gene regulation is similar but more complex in eukaryotes. And then it says something here about repetitive sequences. And we have a lot of repetitive sequences in our DNA that we don't know what it does, but that's part of our genome. Those things are usually used for DNA fingerprinting. So we're going to talk about DNA fingerprinting in topic 19, by the way. So let's talk about what is a gene, okay? So here are some definitions of a gene. You can think of a gene as a unit of inheritance. So often we talk about people having the gene for, let's say, blue eyes or green eyes or brown eyes, right? Or the gene for blonde hair or red hair or something like that. So that's a unit of inheritance, something that we can see or measure, kind of thing. But if you want to think a little bit more about kind of a molecular biology definition of a gene, it's a specific sequence of nucleotides, and it usually codes for something, right? So a gene can code for a protein. So you can see here it's saying coding for a polypeptide. It can also code for an RNA molecule. So for example, we could have a gene coding for a transfer RNA or a ribosomal RNA, and that's never going to make a protein. The RNA is the final product, right? So that's kind of what a gene is. So let's just take a look at, I have kind of a typical gene here, and we're going to come back to this image later. Right now you're going to see how complicated a gene is, and that's the main point I want to make here. So there's the gene, the coding part. It has introns and extrons, and like I said, I haven't talked about what those are yet. So don't worry if you don't know what those are yet. You can always look them up. Before a gene, upstream, we have a whole bunch of stuff. So in this case, these are all regulatory components of this gene. Downstream, there's more regulatory components. So that upstream means before, downstream means after the gene. The gene itself, once it is processed and transcribed into RNA, there's going to be lots of processing that occurs there. And sometimes we have something called a pre-messenger RNA before it gets processed and made into a final messenger RNA. So these are all some things we're going to talk about here in the next few units. Like I said, this unit we're sort of introducing some things, to give you some exposure to it so that when you see it again later, hopefully you'll say, hey, I kind of remember that, and maybe it'll help you remember to figure out how to learn it a little bit. Okay, so let's talk about genes and a little bit of history for you. So 120 years ago, people had figured out what DNA was, and they just didn't know what it did. And so there was some debate, as we mentioned a couple of weeks ago, about whether proteins or nucleic acids were actually the genetic component. But kind of the question is, what do genes do? If you think about cells, cells do the work, or proteins do the work in a cell. So you can see I have some things mentioned there like enzymes or transport proteins and whatnot. So it seems that they're functional. So whatever a gene does, a gene is probably going to somehow make a protein of some sort. So this brings us to this guy. So like I said, about 100 and so years ago, this was, I believe he was a pediatrician, and he was very interested in inherited diseases. So his name was Archibald Garrett. I'm not exactly sure where he was found in the world, but he noticed that some inherited diseases involved misfunctioning enzymes. So in particular, he was studying this condition here. I'm not sure how to say that, alkaptoruria, I think. This is a hereditary condition where there's an enzyme lacking, and in this particular case, urine, it was a blackish color when exposed to air. And he realized that these people were lacking an enzyme, and so he made this hypothesis, which was a huge leap before we even really had any idea what DNA was doing. You can see his hypothesis was that a phenotype was directly related to an enzyme. And this was a very, very good leap in understanding what is going on with genetics. Leap ahead a little bit to the 1930s. And at this point, humans had invented the X-ray. Some people had gotten some cancer. But one thing we knew is that X-rays could make mutants of things. And so there were lots of scientists playing around with these mutants and studying various microorganisms and other things. So these guys were studying a mold called Neurospora. This is kind of a bread mold. You can see probably everyone has seen at some point in their lifetime, and hopefully you didn't eat that bread. The mold is actually very pretty under a microscope, you can see. And so how did the experiment work? Basically, you have some mold, you hit it with some X-rays, and then you can study the mutants that you make. And so in this case here, they were studying the mutants and looking on different types of media. So you can see down here at the bottom, it's talking about putting the cells in minimal media. And in particular, they were looking at something that they realized later was a biochemical pathway. So very similar to something like glycolysis, where you have a precursor. So this was the arginine synthesis pathway. There's the precursor. The precursor is made into an intermediate called ornithine. And then you've got another intermediate called citrulline, and eventually you make arginine. So what they found is that they were able to classify different types of mutants, and I'll show you what that looks like in a chart here. And they were able to make some conclusions around what was going on. So here's our Neurospora, and you can see we've got a bunch of different mutants they're classifying, and you can see that minimal media is a very simple kind of media where sometimes you have to add extra things to it. So they found some of these that could not grow unless they added arginine. Arginine was essential to this. So here's the chart I want to show you. So here's our wild type on the left. And what they're showing here is that this wild type organism, it has all the functional genes. So it grows in the minimal media. It grows in the minimal media plus ornithine, minimal media plus citrulline, minimal media plus arginine, all good. So one of the mutants they called a class 1 mutant, and they found it could grow in arginine, citrulline, ornithine, but not in the minimal media. Class 2 mutant could grow in the arginine, citrulline, but not the ornithine and not the minimal media. So that was a little different. And these class 3 mutants required arginine to grow, but could not grow with any of the other intermediates. So, what were they looking at this? They were able to make this conclusion, right? And what they realized that is that they're looking at a biochemical pathway, and that each of the conversions between the steps in the biochemical pathway were an enzyme. So this was their conclusion, and I'll show you how it works in a moment, but that gene A coded for enzyme A, gene B for enzyme B, and gene C for enzyme C. So the class 1 mutants, they hypothesized had a mutation in enzyme A, class B mutants, enzyme B, and the class 3 mutants in enzyme C. And so they concluded that this was the biochemical pathway they were looking at, and maybe more importantly, they proposed this hypothesis that one gene encoded for one enzyme. We realize now that not all genes are enzymes, not all proteins are enzymes, so this has been revised to one gene codes for one polypeptide. And this is a very important kind of thing to understand about what a gene is, and of course, what's going on with that information in the DNA, and how it's getting converted into some sort of biological function. So, gene 1, gene 2, gene 3, there we go. All right, so test yourself. It says, let's read the question, there we go. Which one of these statements does not describe a gene? So let's read each of these. A discrete unit of inheritance. Okay, so yeah, we said that's fine. We can give it a qualitative definition. A DNA sequence that codes for a transfer RNA. Yes, that's fine. A DNA sequence that codes for a ribosomal RNA. That's good. DNA sequence that codes for a specific polypeptide chain. Yes, that's correct. We just talked about that because that's what Beadle and Tatum did. And E, a DNA sequence that codes for a telomere. No. So if you remember how telomeres are made, they are not coded by a DNA sequence. They are coded by an RNA sequence. So that is not technically a gene. I think that circles that. Okay, so let's move on and talk about getting our information from the DNA to RNA to protein. So I'm going to come back to this diagram again and again and again, because like I said, for the final exam, you need to know all about replication, transcription, and translation. So study those things, know those things really, really well. So in bacteria, all these processes, there's no compartmentalization, so it all happens in the cytoplasm. You got the transcription and the translation. Sometimes it happens kind of simultaneously as well. I think I have a diagram to show you that in a later unit. In eukaryotes, of course, we have the nucleus. So anything dealing with the DNA, so the transcription needs DNA, and so that's done in the nucleus. And anything to do with proteins, the ribosomes are found out in the cytoplasm or in the rough ER. So some of those proteins are translated by soluble ribosomes, and that happens in the cytoplasm. Some of those ribosomes are found in the rough ER, and that's going to lead to membrane proteins and exported proteins. So a little bit more complex, and we're going to talk about those things as well in the next few lectures. So let's talk about transcription. So transcription, this is the process where we are taking a sequence of DNA and we're making a sequence of RNA from it. So if you think about what scribing does, right, or you're basically copying something down, and that's kind of what's going on with transcription. Is the DNA is being used and hopefully to make a perfect copy of RNA. Obviously, RNA has uracil instead of thymine. That's not perfect, but it is really in the same language. It's copying the language of nucleotides to the language of nucleotides. So it's mostly copying. The enzyme that does this is RNA polymerase. So we'll talk about RNA polymerase next lecture and how it does what it does. It's a little more versatile than DNA polymerase, by the way. The second step is translation. So now we're actually going from one language, nucleotides, to a new language, which is amino acids. So this happens at the ribosome. And we'll talk about the ribosome in topic, I think that's 17. We're going to talk about translation and how the ribosome works. So a little bit more history for you. Back in the 1950s, Watson and Crick, maybe you can see them there. Here's Crick right here. And here's Watson right there with the W. Those guys figured out the structure of DNA. So we talked about that in topic 13. And very, very soon after that, people wanted to know how does that information get out? And how does it do stuff in the cell? And how does it make protein? And so a handful of these scientists, and I do mean a handful, it turns out there were 20 of them, formed a little exclusive academic club called the RNA Tie Club. So why 20? Maybe some of you are thinking that number rings a bell. Well, it turns out there are 20 amino acids. So they decided the club would have only 20 members, and they'd meet once or twice a year and talk about this and see if they come up with trying to figure out how does RNA and DNA, how does that information get from the nucleotides to the proteins, right? And there were, believe it or not, to even be more nerdy, they had 4 honorary or guest members, and of course that's for the 4 nitrogenous bases. So kind of an interesting little story there. And anyway, there's Watson and Crick, part of this exclusive club, and they were working hard on this. And so we're talking about late 50s, early 1960s. So they kind of knew this, right? We have 4 nucleotides, we have 20 amino acids. Okay, so we can't just do a 1-for-1. 1 nucleotide equals 1 amino acid. So what if we did a 2-for-1, right? So if we had 2 bases, that gives us, 16 possibilities. Okay, that's not going to work. So they figured that it probably means that you need 3 bases. And 3 bases actually codes for more than enough. That actually gives us 64 amino acids in theory. We only need 20, but they figured it was probably had to do with a triplet of base pairs to figure out the genetic code. So 1960s, Marshall Nirenberg cracked the code, and he was not an RNA Tie Club member. So, all the big shock, he was not as famous as all those other guys. And he figured out actually a very simple way to crack the code. So this is kind of what he did. He made synthetic RNA strands, and he started off with just composed of a single nucleotide. So he started off with U, a whole bunch of U's, right? Added this to a test tube and added a yeast extract. So what's in a yeast extract? Well, if you think about a yeast extract, you're breaking open the yeast cell. It has ribosomes, it has nucleotides, it has everything you need to make proteins, right? And so he basically put in the synthetic RNA and then tested later to see what was made. Probably, I'm guessing, using some radioactivity, but I'm not entirely sure about that. So analyzed the tubes, and this is kind of what he got. He got phenylalanine, 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 a whole bunch of phenylalanines in a row. So he concluded that UUU must code for phenylalanine. So pretty straightforward experiment. Next thing he did, he tried, okay, let's try a whole bunch of A's, and that coded for lysine, a whole bunch of C's coded for proline, a whole bunch of G's coded for glycine. And then after that, it gets a little bit more complex, right? You can imagine all of the other codons, but eventually people were able to crack and solve the puzzle of the genetic code. So there is the genetic code there, and you're going to need a genetic code for your final exam. Don't worry, I will provide one for you, and it will be with the test. So you don't need to memorize it. But let's talk about a little bit. If you take a look, there's one codon in particular that we're going to talk about a lot right here, AUG. So that's the start codon, and the start codon actually codes for an amino acid called methionine. So MET stands for methionine. So every protein, at least initially, starts off with methionine. So there's some stop codons. So I think I have them labeled right there. And so you can see UAA, UAG, and UGA. That tells the ribosome to stop making a protein. So something else about this genetic code is we talk about it being degenerate. So what does that mean? It means that multiple codons can code for one amino acid. So if you look at the example in the middle with proline, CCU, CCC, CCA, CCG, all code for proline. Above it, you've got 4 codons coding for serine. To the left, we have 2 coding for phenylalanine, and so on. So multiple codons can code for one amino acid. Sometimes that third base pair doesn't even matter at all, like in the case of proline. Who cares if you have a C or an A or a G or a U? You still get a proline. In other cases, that third base pair does matter. So sometimes this has a bunch of names. Often in the literature, they call it the wobble position. Kind of a weird name, but it means that the third nucleotide often doesn't matter. So something else that we found about this genetic code is that it is universal, and that means that all organisms usually use the same genetic code. So what's true for humans is true for tobacco and true for elephants and E. coli and all sorts of other things. So that's something else really interesting about the genetic code. It shows that all life is connected somehow. So let's talk about the ribosome for a minute or two here. And like I said, we're going to come back to these things in future lectures. So the ribosome, of course, is where the translation happens. And by translation, I mean synthesis of proteins stringing together amino acids. You can see there's a whole bunch of parts here, all shown in one slide. We've got the ribosome, which has 2 subunits. We've got transfer RNA, and we have amino acids. And so the ribosome is where that translation, converting from one language of nucleotides to the language of amino acids, is occurring. So let's take a look at the transfer RNA. This is the transfer RNA here. And this is kind of a flattened structure of it, and it's RNA. So if you take a look, we've got a sequence of RNA. We've got A, C, C, A, C, so on, and there's a few U's in there. There's a few stars. These stars are just kind of modified bases, so they don't look like the normal ones anymore. And we do have some double-stranded regions in there. You can see these hydrogen bonds. And the kind of most important thing is that there's 2 really important parts of this. One is at the top. So this is where the amino acid is going to attach. So which amino acid? Well, it turns out we have about 20 transfer RNAs. I think there's actually slightly more than 20 coding for the different amino acids. And at the bottom, we have an anticodon. So an anticodon is going to recognize the codon on the messenger RNA. So you can see this one here is AAG. So that would be, of course, 5' AAG to 3'. So that means the codon would be complementary and antiparallel. So it would be UUC, like that. So we'll talk a little bit more about anticodons and codons, like I said, when we get to translation. Here's a picture kind of showing you got the anticodon and matching with some codons. So you can see there's the anticodon U matching with A, C matching with G, and A matching with U. And so the codons, AGU, that's what we look for on our genetic code to see what amino acid that codes for. So U, I'm looking at here, is the first, and then G, and then A. I guess I read that wrong. It's UCA, so sorry about that. Serine. There we go. So that first one is serine. The second one is UAC. So U, A, and C. So the second one is tyrosine. So the genetic code comes in different ways to look at it. I know some of you have maybe taken biology with Trish Collins, and I think she likes this one. You can see you start in the middle and you read one base at a time until you get out to your amino acid. Here's one that's showing just the structures of the amino acids and their functional groups, and some of them have more codons and some of them less. So I'll be using usually the one from the textbook, but sometimes it's easier to squeeze in a different one. So let's talk about how translation works, and I'll show you a problem here in a moment. And like I said, you're going to see a problem like this in the final exam, so it's good to know how to do it. So first thing to do is take your sequence and separate it out into those codons. So codon is a triplet of base pairs. So you can see we got 1, 2, 3, and then I have it separated out. I've made this one very easy. It starts with the start codon. Always look for the start codon, AUG. Remember that one. And then you're going to look on the genetic code to see which proteins they code for. So in this case here, AUG is methionine, ACU is threonine, AGA is lysine, and GGA is glycine. And UAA, there is no amino acid named UAA. So I don't need to write stop. I did write it, but you don't need to write stop. So I'm going to talk about that N and C thing a little bit later, just not today. But if DNA starts off with a 5' and ends off in a 3' end, that's the chemistry around it. In proteins, we start off with an amino group which has a nitrogen, and that's what the N stands for. And they end off with a carboxylic acid group, which is a carbon. So sometimes we talk about the N and the C ends of a protein. So here is kind of what you're looking at, something you're going to look at on the final exam. It's not complete because we haven't talked about everything yet, but this will be part of a question. Is that you're going to be given a DNA sequence and you're going to be asked to give the RNA sequence and give the amino acid sequence for it. So I started this one here. This is a very simple question because the one you're going to get is a little bit more complex. This one I've made very easy because I have the start codon right at the beginning. That may not be the case on your final exam. But like I said, I want to show you how this works to give you an easy example first before we add more information to you. So it says give the RNA sequence. So our messenger RNA, basically we're copying the sense strand, and I haven't told you what a sense strand is yet, so we'll get to that. But you can see we have ATG. So remember, our RNA doesn't use T, it uses U for uracil instead. So basically, I'm copying that top strand, and I'm going to change all my T's into U's. So AUG, CCG, UAC, UCU, AUC, CUA, and UAA. So normally I do this on the blackboard, by the way, but go a little bit slower. So it's worth it for you to work through an example, and maybe what I'll do is try to find an example to send to everybody when we get close to the exam. Like I said, we still have some other stuff to learn and questions and things to ask around this question. So it says give the amino acid sequence. So I usually separate out my triplets, my codons, and then I just need to look at them on the genetic code. So I'm looking up there. AUG is methionine. So hopefully you know that one by now. CCG, so I'm looking up on the genetic code. The first second row, it looks like, is the C is CCG, so that would be proline. UAC is threonine, UCA is serine, AUC is isoleucine, and CUA is leucine. So I know all the codes, 3-letter codes and the full names. You don't need to know all the names and everything. You just need to know what's on the genetic code that I give you. And there's no amino acid named stop, so I can stop there, right? This UAA codes for the stop codon. I don't need to write stop. Okay, so like I said, I will come back to these examples. This is very simple. This is about 2 marks out of 5. We still have to learn about what the other 3 marks are going to be for. So we'll get back to this example. We'll do one in the next unit and the one in the following unit once we add some information for you. So a couple more things to finish up here. I told you I was going to come back to this diagram again and again. And so this is something important for molecular biology because, of course, the genetic code applies to all living things. And so when we're dealing with biotechnology, this means we can take genes from one organism and put it into another for biotechnology purposes. You can see in this case here, they're showing a detergent that has a lipase and a protease in there. So lipase breaks down greases, protease breaks down proteins to help the detergent break things down. And those were put into E. coli for the detergent. Humulin is as a human insulin, also put in E. coli. So this is very, very handy stuff. Some of these biotechnology things are kind of more like novelties. You can see there's a tobacco plant expressing a firefly gene and a pig expressing a jellyfish gene. And we're going to talk about that a little bit more in topic 19 when we talk about biotechnology and genetic engineering. This one here, by the way, when you Google genetic engineering, things like that, you end up with a lot of weird things like this, which are really just somebody with some creative photoshopping abilities. So again, more on this in topic 19. I did want to show this to you though. I think these are really cool. These are called glowfish. I'll play this little video for you.
The glowfish experience is
It'll
unlike
take you the
anything.
volume though. These are basically aquarium fish, and they put fluorescent proteins from jellyfish in them. And I just heard recently you can buy these in town finally. I thought they were never going to make it to Canada, but apparently you can buy them in town. So I thought I'd look into that at some point. Okay, so I wanted to finish off this kind of just relating a little bit to Mendelian genetics. I know we haven't talked about Mendelian genetics, but by that I mean you know, dominant and recessive traits to think about genes. And you may remember that from high school. We've got this idea of dominant and recessive traits. You can see in this scenario here, we've got some sort of flower that's pinkish or purplish, and that's the dominant color, and the recessive color is kind of white. So I have a couple of videos here to show you, and you can see there's our genes. I'm going to press play, and this one here is for the homozygous dominant. So that means that 2 alleles are the same, and this is the dominant color. So let's play. There's the DNA. The DNA, of course, gets transcribed in RNA, and then the RNA gets translated into protein. In this case here, these proteins are little enzymes, and these little enzymes are involved in making some sort of pigment. So like I said, these are the same for both alleles, and there it goes, and it's going to make some pigment. And the pigment is, of course, red, because it's a red flower. And this is the dominant color, so the flower grows and you get red. So what if we have the homozygous recessive? Let's take a look at that. So same thing, we have our genes, our alleles are transcribed into RNA, and RNA is translated into protein. So what is happening in this case here with these enzymes? Well, it turns out in this case, the recessive enzyme is actually non-functional. So you're going to see what's happening when the enzyme encounters its substrate, basically nothing happens. So no pigment is made. So in this case, no pigment is made, we get a white flower. So what happens if we have heterozygous? So that means we have one allele of the functional enzyme and one allele of the non-functional enzyme. So in this case, let's watch and see what happens. So there's our RNA. The RNA is translated into protein. Proteins fold up. One of them is going to be a functional enzyme and one of them is going to be non-functional. So let's see what happens here when the pigment is made. Are we going to get pink? Are we going to get red? And it looks like we have just enough to get a red flower. So in some cases, genetics is a little bit more complex. You might get pink flowers or different, different shades in between. Genetics is not quite as simple as Mendel had determined, but in many cases, this is what happens. And so you're really looking at a gene, which is DNA, which is coding for a protein, and the protein leads to some sort of phenotype. Some phenotypes are very visible. Like I said, hair color or flower color or something like that is very, very visible. Some phenotypes are a lot less visible. Okay, so that is actually the end of this topic. Basically, like I said, it's an introduction to the next few topics where we're going to talk about what's going on with that RNA and how those proteins are made, how those amino acids are strung together. And we're going to talk after that a little bit about how genes are regulated. And then the last two topics, we're going to have one lecture on biotechnology and forensics. And then we're going to have about a lecture and a half, maybe 2 lectures on viruses. And we're going to talk a lot about COVID-19 and coronaviruses. So hopefully you enjoy that when we get there. So that's all for today. I will see everybody on Wednesday.